So, Sai, what is genetics?
Genetics is the study of genes. Yes, I am being deliberately obtuse.
Yeah, yeah.
Okay. So I I am Kieran Stevenson or Syralth or Sai. Sai is usually easier. I'm going to basically talking today about like an introductory lecture to genetics for everybody. As I'm going through this, please do speak up if you either don't understand my accent or I've missed something or you want to ask a question or whatever. So you can either speak up with the audio or use the chat and somebody will poke me because I don't have the chat on my screen. Um, thank you, Windows. So just a little bit about me and why I'm qualified to talk about this subject. I'm the scientific director at the FFF, and I kind of help out on the board and help decide where we go. And I'm going to be as vague as possible. At the moment, I'm a postdoctoral researcher in Cornell, and I'm studying antibiotic resistance using the CRISPR protein, which you might have heard in terms of gene editing. So we do a lot of research with that. And Cornell are the one that gave me access to Zoom to do this, which is nice. So, what we're going to cover today, we're going to cover like 5 topics. The central dogma of biology, DNA to RNA to protein is kind of how most of life works. Then we're going to talk about what DNA and genes actually are. We're going to talk about gene regulation, how we can turn them on and off and decide what the cell actually does. DNA packing, how the cells hold it all, and by it all, I will explain in a minute after I finish disassembling my keyboard. And then I'll give an example of how kind of gene regulation all comes together, a nice simple one that gives you an idea of how things work. So kind of the first thing to know about biology is it's complicated. You might have seen this diagram of a cell in like high school. So we've got this dark nucleus with the DNA inside. We've got the cytoplasm where most of the chemistry of the cell happens. We've got the mitochondrion, which is the powerhouse of the cell, as everybody likes to quote to me, because that's the one thing they remember. And it's all enclosed within this cell membrane, which is kind of layer of oil and fat that surrounds the cell like a bubble. So it's actually quite soft. But this is what you're taught in high school. This is what you end up getting taught in like early years of undergraduate biology. There's a lot more stuff in here. And often, you get like one example of something when in fact there can be hundreds and thousands of these different structures within a cell. So it's actually really packed full of things. And then you get on to, if you happen to be a biochemist like Atta here, you might actually learn about the individual chemical reactions that go on in a cell. This is just a small portion of what goes on in the cytoplasm. This is just how you take in nutrients and convert them into something else or convert them into energy. So there's just lots and lots of chemistry here. Fortunately, I won't be talking very much about this at all because we will be here forever and I really don't want to be. The other thing to know about biology is it covers a wide range of values, so you get used to seeing very big numbers and very small ones. So for example, number of cells in the human body is about 3 times 10 to the 13, that's 30 trillion cells. You've got about the same number of bacteria, most of which are in your gut. The reason you just don't notice it is because they're a lot smaller. In terms of how much DNA we've got, we have 3 billion bases. That's 3 billion letters in our genome. And that's in every cell. Fortunately, the size of them is they're really, really tiny. They're about a billionth of a meter. But because we've got 3 billion of them, it means we've got about 2 meters of DNA in every single cell in our body, which is quite a lot. So if you were to extract all the DNA from somebody, they would die. And if you played it end to end, it would go from the Earth to the Sun and back about 200 times. So this is a lot of stuff crammed into what is a very tiny space. But for the sake of our heads not exploding, we're going to simplify. This is as complicated as the cell I'm really going to talk about. It's going to be a blob. We're going to have a blob, we're going to have a nucleus and a cytoplasm. And we'll ignore the mitochondrion because currently we're not interested in power. So, the central dogma of biology, this is kind of how everything works and kind of the basic idea, is you have your DNA over here, and the DNA of the cell contains all the genes that an organism has. And what you want to do, what happens is you take one gene and you copy the information into an RNA strand. The RNA strand is then turned into protein. And don't worry, I'll explain what all these things mean. Basically, proteins are your most common bit. They're the thing that gets things done in the body. They fall into some general classes. There are things like structural proteins. You've got this tubulin, which forms what's known as the cytoskeleton, basically the skeleton of the cell, that gives it structure and rigidity and allows things to be transported along them. You get signaling proteins, so if this is the cell membrane, the signaling proteins are here in purple. They receive signals in the form of chemicals from outside the cell, and then they do something with them. Sometimes it's a chemical reaction, sometimes they send off another protein, like this little green one, off to do something elsewhere. And then finally, we have enzymes. These are the bulk. These are the guys that do all that big chemical processing I showed you earlier. So for example, this guy takes this substance and splits it into two products, and then it goes off to do the same reaction again. Basically, every enzyme does one specific tiny reaction. So for every single one of the arrows in that complicated diagram I showed earlier, there has to be an enzyme that does that thing. So it means there's a lot.
We can add that some enzymes do have a bit of a more complicated mechanism to them where they might need to do two reactions at once, but they're rarer, aren't they?
Yeah, they're rarer. They're more complicated, and we could spend all day talking about we could spend all day talking about the exceptions to things. So the information for all those different enzymes and stuff is all stored in the DNA. And what we want to do is extract them one at a time and turn them into protein. So we make the enzymes one at a time so we can control how many they are. And the thing about DNA is it's kind of precious. Like I said, it contains all the information and genes that an organism has, and you only get one or two copies of any given gene. And you don't want your proteins all the time because, well, a brain cell has no need to digest fat, for example. And you might need a lot of a given protein. So things like those structural proteins, like those microtubules, need an awful lot of them in order to be able to make the cell more rigid. And if you break your DNA, you probably can't fix it, which means you kind of don't want to do too much with it. And particularly when you consider that proteins and the building blocks are quite reactive. You want them to do chemical reactions, but that means they could damage the DNA. So you want to keep them, and some of them you want completely out of the cell. You don't want the enzymes involved in, say, digesting your food anywhere else in your body, because then you will get quite ill. So sometimes you want to produce them and kind of shove them out of the cell immediately. And definitely away from your DNA. So what happens is the DNA is kept inside the nucleus, inside that blob, blob inside the bigger blob. So in this blob inside the bigger blob. So we've got the cell here in this outer membrane, and then we've got the nucleus, and this is where the DNA is stored, this kind of twisted blue and red thing here. And what happens is this protein called RNA polymerase. I'll explain just what that does in a minute. But it basically produces this RNA. The RNA carries information from this protected region where the DNA is out into the cytoplasm, where the factory of the cell, this ribosome, can build it into a protein. So the ribosome does the fancy translation of the letters into chemicals. And basically, all I want, if you're going to take anything away from my rambling, basically all genes follow this pattern. You go from DNA to make an RNA to make a protein, unless you happen to be a virus, or you're making an RNA gene, or you're part of the ribosome. Basically, biology has a lot of exceptions. Like I mentioned, there are enzymes that need to do 2 or 3 reactions at the same time. We could be here all day if we wanted to explain the exceptions to the rules. So basically, this is kind of like what I'm giving you today is kind of the general overview, what most things do, but there will always be exceptions, and biologists love to point them out. So don't, Zenny.
I've been holding myself back for like 5 minutes. Yes. But yeah, it's really good.
I am oversimplifying, and I am leaving out a lot of exceptions.
No, you're good. You're good.
Sorry, I just checked the chat there. Yes, it is oversimplified, and I am. So what is DNA, RNA, and what are genes? Because you tend to discuss them in terms of you get genes from your parents, etc. But we need to know what they actually are in order to really understand how they work for what we need here. So DNA is deoxyribonucleic acid, which, unless you know your chemistry very well, will not mean very much to you. What we all recognize, though, is this kind of twisted ladder structure. We've got the kind of outside of the ladder, then we've got the rungs across, and it's all just twisted slightly. And what the outside of the ladder is made of is the deoxyribose sugar. So we've got these sugar groups here, these little pentagonal things. They're connected together by phosphates. And this forms a really strong, rigid backbone. But the bit we're all interested in is the letters, which I'm sure you've seen, the A, T, G, and C. And these are called bases. And these form up into pairs. So we've got the A, the T, they always bond together. We've got the G and the C, they bond together. But these bonds here, they're just hydrogen bonds, they're quite weak. So I have an example here. It's stuck to things. It's a bit like Velcro, in that you've got this really strong, rigid backbone. You can't actually, generally don't snap your Velcro, but you can pull it apart when they're stuck together quite easily. This is kind of like DNA. So they've got these very weak bonds in the middle and a very strong backbone. And the reason I'm trying to explain, the reason I'm explaining this is that because it allows you to copy the DNA without damaging it quite easily. So when you're wanting to make RNA or you're wanting to make a new bit of DNA, what you do is you take your DNA, split it in half, and then you have a protein called a polymerase, which comes along and looks at every base and tries to pair it up with one that's kind of floating around in the medium, that's floating around in the cell. And so it makes a match. So between T and A, there's a match. The polymerase puts it in, attaches it to this growing chain. And this is how you build a new strand of DNA when you want to, say, divide your cell. Or in the case of what we're interested in today, this is how you make RNA. It's just, instead of grabbing a DNA base, you grab an RNA base.
Yep, and it takes off the 2 spare phosphates, which have been used to kind of keep those free-floating nucleotides from attaching to the wrong thing.
Yeah. Yeah, so they also provide the energy to actually do the whole chemical reaction.
Yeah.
But we're not going to get too much into chemistry. We'll be here a while. So, like I mentioned, DNA has bases. We generally just shorten these to E, T, G, and C. They always form pairs. The only exception is when you're looking at RNA, because it's slightly different chemistry, you have a U instead of a T, which is just lovely and confusing when you're looking at transcripts. But basically, but they pair up. So, for example, the polymerase has made RNA of this top strand here. So you see the A matches the U, the T matches the A, the G matches the C, and so on and so forth. We could do this for all 3 billion bases in the human genome if you fancy. No? Nobody interested? Okay. So Okay, so we have letters in a row. How does that get us to actually making chemicals that do things? And this is the genetic code that tells you how to turn these letters into actual chemicals in a chain that make a protein and do things. The way this works is you look at three of these bases at once, and this tells you which amino acid it is. And this is what this diagram over here is trying to tell you. It's not as complicated to read. Basically, you start in the middle. Like for example, for this codon, you start in the middle and you go U, A, and then there's a C here, and then you know to attach this protein. I'll talk about the machinery that actually does this in a second. There are two important things to note on the diagram. There is this one here, which is only one sequence that matches it. This is methionine, and this is always the start of a protein, is always this. And the end of a protein always comes as a result of this terminator sequence, this TER here. And this basically is saying where to start building your protein, where to stop building your protein. And the thing that actually does the building of the protein, I mentioned it earlier, is the ribosome. So what the ribosome does is...
Sai, there's a little question for you. So any two letters can pair up?
The two letters only pair up as in A, T, and G and C. So A is only bond to T's and G is only bond to C's. You see they've got slightly different shapes in this diagram, so you can't accidentally mix them up.
Dreamcloud, did that answer your question?
If I can speak. What I'm confused is if the wheel, why does each section of the wheel make it look like the two letters can pair up if it's only one letter compared to another? Like it looks like A can pair up to A.
This you read it in the order. So for I don't have 2 pointers. This is annoying. So you would read the U here, and then you read the A next to it. So instead of being paired up, this is actually just joined together in a line. Does that make sense?
To kind of explain it with different words, the wheel on the right is just a visualization. So the very center is your first base, then the kind of the middle area is your second base, and then at the very edge is bases that are options. Any one of those options can be used for the third base of the codon.
Yeah, sorry, I didn't explain that again. So there you go. So there are certain letter groups that will code for the same thing. Because there were only 20 amino acids, but there were 64 possible combinations if I got my maths right. Probably not. Somebody check my maths. So the thing that actually does this is the ribosome. It's the big factory of the cell. This does most of all this translating. And what it does is it finds your RNA and then scoots along until it finds this first methionine, what I call the start. Then it recruits the bigger part of itself. Because
it comes to complicate this.
Yes, it's starting to get complicated. Yeah. So basically, it scoots along to find the start of the protein and then starts building on from there. So we've got so this is the ribosome. It scoots along and it looks at 3 bases at a time. Just ignore what this is for the moment, and then adds the appropriate amino acid to the end of the chain. There's a lovely video of it. I'll link it later. I don't want to get copyright because I can't reference it. But to go back to this, to sum it all up, we've got the DNA, we've got the polymerase, which makes the RNA by matching up the bases in their AT and GC pairs. The RNA is transported to the ribosome. The ribosome does the fancy decoding and makes the chain of amino acids, which make your protein, which does things. Hopefully useful things. Now, this has all been talking about sort of like one gene. You might want to know how many we have. So in the human genome, which is all the DNA a human has, we have 20,000, about 20,500. It varies depending on exactly who you talk to and which data you use. About 20,000 genes, and our genome is about 3 million bases long, so 3 million of those letters long.
Was it? Are you sure it was 3 million bases? I thought it was 3 billion. Am I getting mixed up?
It was 3 billion. Give me a second. Oh, it's going to be fun for the edit.
Google says 3 billion.
It was 3 billion. Sorry, my mistake. My typo.
No problem.
Okay. So
what's a few orders of magnitude between friends?
It's one letter. So it's very, very long, but only about 1% of it actually is part of these genes that make protein. It means there's a lot of it either doing something else or doing nothing. About 44% of it is made of transposons. These are bits of DNA whose only job in life is to make more of themselves in our genome. They're like little viruses that kind of hung on. About 10 to 15% are actually gene regulation. So they're about switching these genes on and off instead of making proteins themselves. And another 10% encode for kind of like more general regulation mechanisms or control sequences with kind of like a distant action. So they're not associated with a particular gene, they're kind of associated with broad stuff. And I can't get more specific than that without just listing examples at you. But it basically means about 75% of our genome isn't really useful for us. It doesn't do anything that makes us live.
Can I say that doesn't make it junk? It just makes it not directly used in the cell's sort of normal chemical reactions. But it might still be involved in like the structure of the chromosomes, for example.
Yeah. We'll get onto the structure of chromosomes later. But yeah, so a lot of it is not doing very much. The reason I point that out is because there's a lot of space we can add more things, and I'll explain why this is important in a minute. The other thing though is that in order to actually build a human, you need somewhere between 100,000 and 50,000 different types of protein. Now we've only got 2,000 genes, so there has to be some way of getting more protein out of them than it would look like. And this is where alternative splicing comes in, which is lovely, which is biology making use of junk in our DNA. Specifically, that a lot of genes in higher organisms generally have basically junk stuck in the middle of them. So the bit that actually makes your protein in this diagram here are your exons, and then you've got these effectively non-protein or non-useful bits of DNA stuck in between them. And what the cell has to do is basically, after the RNA is made from the DNA, because it's copied exactly, it's filled with junk, you have to actually cut them together in order to build your protein. Like, for example, there's protein A that's cut out the junk, so you've got your nice single protein here. But what the cell also does is it will sometimes skip. It can skip these exons or shuffle them about in order to get other proteins from effectively the same stretch of DNA. So, for example, this protein B is missing this exon 3, and protein C is missing exon 4. And this will tend to happen in different cell types. So, like the brain will maybe have protein A, your muscles might have protein B, your skin might have protein C. This allows the cells to basically get lots of different types of things out the same stretch of information. So just to summarize, the human genome you've got about 20,000 genes producing 100,000 to 500,000 proteins. But in general, you're looking at a single gene, which is turned into an mRNA, which is turned into an amino acid using the ribosome and using this 3-letter codon system. Now, when we get into gene regulation, you probably have some questions. Like, if most of your most of the DNA is junk, or at least not making protein, then how do you know where to make your RNA from? So how does the polymerase know where to land in the DNA to make it the RNA? How do you know how does it know which gene you need for a given cell? And what if you only need some of them some of the time? Like, for example, you only need to make digestion proteins when there's actually food to digest. And this is what cell regulation does. So we have these lovely diagrams. Yeah, we use colored blocks because they're simple to draw and we like Lego. It looks nice. So what we've talked about so far has mostly been these blue sections here. So this blue section is the bit that actually turns into mRNA. We've got the exons that actually make the protein and the introns that are cut out. And in order for the polymerase to land, it recognizes something called the promoter. And so this is where the polymerase lands and then basically starts doing its making RNA and works its way through the gene here. The promoter can have different strengths. So if it's stronger, it means the RNA is more likely to bind to it more often, and therefore you get more expression of your gene. The distance from the start of the gene can vary. So there can be space or things to get in the way. And I'll talk about those things in a minute. And there is a direction. So it doesn't matter in human genes because our genes are very far apart. But in things like the viruses, they can actually have genes overlapping each other. And the way they do that, basically sharing the same bit of DNA. And the way they do this is they have the promoters pointed in different directions. So if you go one way, you get one gene. If you go the other way, you get another gene. And different species have slightly different types of landing sites, which can make it quite tricky if you have an unknown organism to figure out what gene it actually is. Okay. Now, the promoters are fixed based on the sequence. So based on the sequence of letters in the DNA, you can't change them. So you have these things called activators and repressors, which allow you to actually turn the gene on and off in response to a signal. These are proteins that respond to some sort of signal, usually a chemical. They will land on the DNA, and they can either, in the case of activators, hold on to the RNA polymerase and help it get started. Because the RNA polymerase is a bit like an old car. It takes a few minutes. It can take some time for it to actually start doing its thing and moving on the DNA. Until then, it might fall off. And so the activators can help hold the polymerase, either bring the polymerase to the DNA or help hold it on until it starts doing its thing. Repressors do the opposite. They switch the gene off. They can either push the RNA polymerase away, or more commonly, they tend to sit on the promoter or between the promoter and the gene, basically running like a massive roadblock to stop the polymerase doing its thing. And depending on what you need your gene to do, you can have any combination of these. So you can have several different activators, several different repressors. You've got similar elements, like these are distant from the gene, so these could be quite a long way away from a gene. These are called enhancers and silencers, and do very much the same thing as activators and repressors, just from a longer way away. The usual way this is explained to be accomplished is that because DNA is very long and relatively flexible, is that the DNA curls back and allows these distant enhancers, etc., to interact with the closer elements and what happens. They can be a little harder to identify though, because they are so far away, it can be harder to pick them out. You just look at a gene sequence. Yeah, finally, the last little thing is you need to stop the polymerase at some point, because otherwise it will keep running through your entire DNA, and that won't be great for anybody. So after transcribing the gene, the polymerase starts with the promoter, works through the gene, and then encounters this poly-A signal and the terminator. So the poly-A signal is a long chain of T's in the gene. These become A's in the RNA because T's and A's pair up. And either the polymerase itself or other proteins around recognize the sequence being produced and go and pull the RNA polymerase off the DNA. This lets the mRNA be able to be transported out. out of the nucleus and off to do its thing. For some reason, I forgot to put an image here. But when it goes to do its thing, you can also regulate it as well. You can also regulate it after it's out of the nucleus. In this case, you can regulate the protein synthesis itself. Since the ribosome needs to find the mRNA, it has its own landing site, a bit like a promoter, from where it starts and then moves along the mRNA. And again, these can come in a range of strengths. You can either have really strong ones, so from a small amount of mRNA, you get a lot of protein, or you can have weak ones. And you can also slow the ribosome down with certain sequences. So certain rare amino acids for your chain, and have to wait a while for it. Wait a while for an amino acid to come along so it can move on to the next step. This allows you to do things like change the time dynamics. So if the ribosome is moving slowly, then you've got your protein being expressed for a relatively long time after you've activated your gene. Or you can make it really quick. And finally, you need to get rid of your RNA, because if it just hung around, then your gene would be on the whole time. So this is what this poly A tail I was talking about at the beginning. So we have the mRNA, and it has this tail at the end. And then this enzyme called deadenylase comes along and slowly eats away at it. And this can be sped up or slowed down by different types of proteins that will sit on the tail and get in the way. And some of them can add more A's on, etc. But basically, it works kind of like a timing signal. So this is slowly eaten off over time, and then once it is gone, these exonucleases, which are really fast, will chew up the mRNA. And the reason these have evolved to be very, very quick is because viruses often try and inject their own mRNA. So they recognize that, oh, it doesn't look right, I'm going to eat it, which is actually how most of the immune system works. So to summarize, because that was probably a lot, there are lots of different ways of influencing gene expression. So you can change the promoter strength, you can have activators or repressors, and enhancers and silencers that turn on and off in response to signal. You can change the ribosome binding rate and the speed at which it moves. So you can adjust how much protein you get from a single activation of your gene, and you can adjust how long this RNA hangs around. So how much protein it can make over its lifetime.
Hey, Sai. Before you go on, you mentioned something really interesting that I just want to maybe get you to speculate about, if you could. Like your mention of the 5' exonucleases being evolved primarily to like counteract viral DNA, or at least like being involved in the process. Do you think that that virus stuff has helped like evolve how eukaryotes work?
Yeah, there's a lot of suggestions for that. So things like the transposons in the DNA and the exon, like when you're splicing together those exons to make the transcript, you're getting rid of this viral DNA that has landed in the your gene. Some of these bits are definitely viral origin, and basically our bodies just learn to cut them out. So there's definitely quite a few places where there are things like that. So,
yeah. Well, bacteria have definitely helped eukaryotic evolution.
Yes, things like a lot of your nucleases and things are designed really to have evolved from where bacteria deal with viruses and stuff, and they definitely target like restriction enzymes and stuff are almost always to try and get rid of viruses.
That's really cool.
So,
hold on. Another thing. I don't know if you're going to get as far as mentioning, but there are other ways as well that that gene regulation can occur, so that you get different uses from the same genes and different proteins out of them.
Yes. So like I said at the beginning, this isn't exclusive. This is just a general process. So most genes will fall into this pattern. It's not exclusive.
Yeah.
There are cleverer ones.
Absolutely. There's some variables, for example, in how the ribosomes actually translate the mRNA and make a protein from it.
Yeah.
So that comes into play too.
Yeah, the number of proteins we'd have to talk about to get into that would get a bit ridiculous. I looked at it and the diagrams get quite ridiculous because it's very complicated. But yeah, so there is stuff here I'm not talking about deliberately because it's either too complicated or will be here forever. Like I said, biology likes to break all the rules. So DNA packing, to go back to the previous section, you have to shove 2 meters of DNA into a single cell. You can't just let it hang around because otherwise it's going to get tangled just like my cables in my box over there. So you need some way to organize this. And we have these things called histone proteins. There are 4 different proteins and they form together into this octamer, which is basically just this 8 proteins folding together into this nice disc shape. And the DNA wraps around these discs and you get this kind of a beads on a string, which is basically what it looks like under a microscope. And this helps tidy it up. The histones, after they've got the DNA wrapped around them, can pack together even tighter. So if you look on the left here, we've got the DNA wrapped around the histone, so you get the beads on a string. These histones can then pack together to make these thicker fibers. You can then get further proteins that kind of build a scaffold and assemble it together further and more. And especially during cell division, chromosomes get really packed and really dense. And this is what makes your kind of class, if you've seen them at high school, these classic dense little black X's and things you see in the middle of cells. We're not going to talk about them. They are really complicated as to why that is. But active genes tend towards the left here. They tend to be quite open. This allows all the proteins we've been talking about to get at the DNA. Whereas inactive genes tend to be packed up quite tight and effectively tucked out of the way. So that's why I mentioned packing, because it's important in gene regulation as well. So yeah, there's more levels of control. So the histone proteins themselves, they aren't just a disc, they're disc with little tails hanging off, like you see over here. These tails tend to block access to the DNA. And what some proteins can do is they can add acetyl groups to it, which is a 2-carbon acid. It's not too important exactly the chemical structure, but basically when they add it, these tails open up, and this allows proteins to get at the DNA and be able to do all their other gene regulation stuff on it. They also attract these bromoproteins. These proteins attract the these proteins also help attract all the other parts of the gene regulation, so this can make it even more active. On the other hand, you have methylation, which is a methyl group, which is a single carbon. If you attach that to the tails do change shape, and this allows the histones to stick together better into those fibers. And from that, you can recruit these chromoproteins, which cause the fibers to build up into those big scaffolding structures. This again lets you to shut a gene off really, really off. And this is so this is kind of the last stage I'm going to talk about here in terms of turning things on and off. So what I'd like to do, to finish up, is just kind of give one example of how how one gene is turned on and off. Now, when I started this presentation, my example was intending to be in humans. The problem is humans are quite complicated. You might have noticed we're quite complicated things. So, for example, if you were to try and look at genes involved in, say, cell division or growth or death, they're all linked together and they all take multiple factors. So you can have chemicals like hormones or growth factors or some component of where the cell is. They feed through these transduction proteins, and then they activate other proteins that have lots of other effects on each other. This is called crosstalk. It allows multiple signals to do the same thing. And then they do things. But basically, I can't give you a solid example of this because we'll be here forever and I'll be listing 3-letter proteins that won't mean anything to you. So we're going to go back to the classic example of biology. We're going to take an example from bacteria, and it's going to be the lac operon, which you might, if you've done biology, you'll have heard of. Basically, lactose is a useful sugar. You'll know it from milk. It's like a kitchen to a really big restaurant. Yeah, it pretty much is. So lactose is a useful sugar. When it's present, the bacteria wants to eat it. And we have 3 proteins that do this. We have this lac-Y, which pulls the lactose into the cell. Lac-Z, which breaks it down into nice sugars. And then we've got this lac-I, which actually detects the presence of lactose. And we'll ignore lac-A because that's chemistry that I don't want to get into. But basically, you don't want to be wasting time making these proteins if there's nothing there to eat. So how does the bacteria turn it on and off? Okay. So if we look at the diagram of the actual gene in bacteria, ignore the fact that there's 3 proteins right next to each other. That's just how bacteria work. We have this lac-I. We have a CAP binding site. We have this P, which is the promoter, which is where the polymerase lands. We have this O. And we have our proteins we need to actually make use of lactose. Don't worry, I'll explain what the letters mean. There we go. So, for example, in a situation where there's lots of glucose available, which bacteria love to eat over lactose, and there isn't any lactose, what happens is this lac-I is produced, and it sits on this O site. And basically, if the polymerase was to try and land on the P and move through the genes, it can't. It just run into the repressor protein. On the other hand, if there isn't any glucose, so the bacteria is looking for alternative food sources, the CAMP protein gets turned on. It lands on the chromosome and it tries to bring polymerase along and do its thing. But because this repressor protein is still in place, because there's no lactose, it can't do anything. So it's still switched off. On the other hand, I got those out of order. On the other hand, if there is lactose available, this protein falls off, which means the RNA polymerase can do its landing thing and then go through these genes. Once it does so, you get your mRNA. And this AUG is this methionine start. We've got the stop, which means the ribosome can land and make its protein. And because there are several proteins on the same bit of mRNA, ribosomes will produce all 3. And that's how bacteria turn on. That's how bacteria start digesting lactose if it's available. And this is kind of like nice, simple how genes are switched on. Because we've got this CAP that activates only when it needs this sugar, and we've got this repressor that only deactivates when there's actually lactose to eat. And yeah, and so just to wrap everything up, in conclusion, it's complicated. As Rosie said, it's a kitchen to a really big restaurant. There is a lot of controlled chaos going on. It depends mostly on what the sequences are in the actual gene, as to like activators and repressors will look for particular sequences to land on and do their thing. You can turn this regulation on and off with signals from many different places, from outside the cell or from inside the cell, or by combining different signals together. And so for what we care about in the FFF, if you want to add a new gene or alter a current one, you kind of need to take all this into account, because if you're changing anything about the gene, you could change a lot of these different stages in the process, and it's important to know what they are. And also, for genes that already exist, sometimes we want more of them, sometimes we want less of them. I would love to have more fat-burning enzymes, for example. Unfortunately, we haven't figured out how to do that without really bad consequences. So, yeah, this is the kind of thing we need to look at if we want to actually know what we're doing. But hopefully I've given you some idea of kind of how in the simplest way, kind of how things all work. So does anybody have any questions?
I have a tangential question. It really is.
I like tangents. Tangents are good.
I'll admit a lot of stuff went over my head. I kind of understood some things. So being a computer programmer myself, a lot of it seems very computer-like in the DNA. And I'm aware that there's not a study, but they were able to store, or maybe it's just theory, store data into DNA as like a separate way to store data.
Yep. Yeah. So so the way they did that was just by basically creating the gene, using the letters that already exist as a kind of, instead of binary, it'll be quaternary, and you can use that to encode information. In terms of actually expressing genes, it's so the way I was talking about it is you can switch them on and off, but it's kind of a probabilistic thing. So if you actually look at the level of expression, it's kind of, you get this kind of general curve. I will get a pen up on here because otherwise I'm going to be trying midair. So if you're writing with a mouse doesn't work. So if you were to look at the amount of gene you get for some sort of activator A, if you increase the level of A, you kind of get this kind of general slow rise, and then there's only so much amount of gene you can get. And if you were to look at, say, like an activator protein, so the amount of activator protein activated in, say, like the no lactose and then the lactose, the amount of activator protein kind of follows this kind of broad distribution. So it's never really on, never really off. There's always some little bits that kind of hangs over the line. Those graphs aren't the best. I apologize for that.
You're saying it's analog.
Yes, so that's a better way of putting it. Thank you. It's analog, not digital, which means the computer analogies start to break down once you look at bigger and bigger systems.
Yeah, there are additional complexities as well. So one thing is there's a huge focus on context, like other proteins or other molecules that are present in the system that interact in really complex and poorly characterized networks. And this thing about the was it like the RAS-RAF pathway and stuff, like that is even still like a gross like simplification of the system. Like all those things will crosstalk and who knows what, and you got splice variants even of those, and like who knows what. So one other thing, like one additional source of complexity is localization of these molecules matters a lot. So the localization of the RNA, depending on where it is in the cell, will greatly affect how it's translated. The localization of proteins in the cell will likewise greatly affect how they function. Sorry, go ahead, Sai.
Yeah, the other thing to remember is it's in a computing system or like in engineering or physics, you generally are able to do like this problem in isolation or look at this thing in isolation. When it comes to biology, like you have the nucleus and then you have the cytoplasm, and it's just a big chemical soup. Everything interacts with everything else at some point. Maybe not in a deliberate way, but they'll bump into each other, which means changes to any kind of chemistry and things will have knock-on effects on everything because you can't really isolate stuff. So when we've tried to build like gene circuits based on like actual electronic circuits, it very quickly breaks down because our transistor over here starts talking to the transistor that's in a completely separate gene just because they look kind of similar and the chemicals happen to bump into each other. Yeah. Biology is annoying. It's really hard to pull things out and isolate them. We're working on it, but it's annoying.
We'll have to encode genes that build like little compartments around the different genes.
Yeah, and there's the issue of making that stable.
Yeah.
And I'll pull up that video actually. It's kind of a nice demonstration. I don't know what you guys can see.
We can see your PowerPoint and you're scrolling through the side of it.
Yeah. Can you see my browser?
Nope.
Okay. I'm just kidding.
You might be specifically sharing the window.
That's what I'm thinking. Give me a second to switch screens. So this is the video I wanted to show. I'll just mute it for a moment. So this is the mRNA, and this is the ribosome doing its thing. And you see this sort of jiggly, wobbly chemical soup. And this is a nice way of looking at it. And this is the protein coming out of the ribosome. So you've got these kind of blobs of atoms and the protein coming out and coiling itself up. So you see it starts coiling up as soon as it comes out. So there's sort of this chemical soup.
Unless there are chaperone proteins available to hold it open.
I wasn't going to mention that. We could, like, if you were to talk about all the other proteins involved, you will be here a while, because there's about 16 involved in just getting the ribosome onto the mRNA properly.
And I assume this is talking about only the soluble ribosomes.
Don't stop that.
Yeah.
So this is the ribosome actually reading these individual three base pairs one at a time.
Do you think that when the ribosome reads that it has any reading comprehension?
I'm going to smack you. You know exactly how this works. You're a biologist. Yeah. Well, welcome to our usual board meetings. It's like a lot of serious stuff and then bad jokes. Mostly bad.
So, just to return to something Zenith was talking about, it does matter whereabouts in the cell your mRNAs are, and the proteins are being made from them. A particularly obvious example would be a neuron, because of how long it is. You can have very different chemical conditions at one end of the axon of a neuron compared to at the main, soma, the nucleus blob at the top end of the neuron.
Yeah. You can even have it, even in that blob-shaped cell, between the cytoplasm between right near the nucleus and right near the surface is actually different enough that chemical reactions behave differently. And the cell actually exploits that to do certain things. Yeah, welcome to biology. We could keep going all day. There's a lot of this. There's a library of things we can keep adding to this. Any specific questions or...
I don't really have a question, but I do want to kind of point out that I think that in terms of maybe keeping this going or like going into other subjects, I think that, well, embryonic development is still kind of a love of mine. I think that could be like really cool.
You're in your room that one. I hate embryonic.
Well, you hate embryonic development?
I hated it as a subject to learn because it was very much this does this to this to this. And I end up very bored trying to memorize these things. I much more enjoyed the chemistry and the sort of the building blocks and then using them to do something, whereas developmental stuff tends to be a lot more scripted. It's like I hated signal transduction because this protein interacts with this protein and does this protein.
Fair enough.
Welcome to my plate. You can do that, yeah. One thing that we possibly should cover is, now that I've done genetics, possibly genetic engineering.
Genetic what?
Genetic engineering.
Oh,
really?
Yeah.
I think really simple bacterial stuff though, because getting towards humans is very complicated very quickly. But we could do
Still didn't hear you properly. Sorry, genetic what?
Engineering. You could do
engineering.
Do things like probably a simple one would be plasmids in bacteria, and just basic kind of restriction enzymes and putting together a gene circuit, putting together something like the lack of one, because that's always used in genetic circuits.
So you would want to talk about the current state of the art with that?
Not really. I would mention things like CRISPR proteins, etc. But if you talk about the state of the art, you need to have the grounding in actual genetics in order to understand why this is important.
Yeah. Any other requests?
Well, I sort of have another tangential question, if there's nothing else.
Josiah Zena glow-in-the-dark yeast kits? I do not know him, but I'm not surprised.
I know about him.
I'm not surprised. I haven't necessarily needed them because I've got glow-in-the-dark stuff in the lab.
Yeah, I'm aware of Josiah Zayner and what he's been up to. I think he's a little bit on the irresponsible side in some ways, just in terms of the approach he's taken has been a little bit sort of like try to edit genes first, ask questions about what the hell the genes are going to do later.
Yeah,
because he didn't really approach it from a point of view of actually understanding what all of the effects are going to be. And that slightly worried me. But
so that's kind of the motivation.
He chose some gene edits that were very simple, and he was doing yeast, which is more understood by science than humans. But you know, then he was also, I think, I'm not sure if it was him or someone else that was trying to edit their own genes as well.
Yeah, people tend not to yell at you if you experiment on yeast. When you get to humans, they tend to yell more when things go a bit screwy. Nobody minds if you kill yeast. If you kill a human, you're in trouble.
Yeah.
Part of the reason I was kind of motivated to kind of do this seminar is just because it's really complicated. You think it's as simple as taking a gene from, I don't know, from like a fox or something, putting it in a human. There's a lot of this kind of tinkering with very complicated systems you have to do to make it work.
Yeah, I mean, you know, the
Not
examples
that you can't do it, just that you can't expect to do it in your garage. Yeah.
The examples that we've got so far of people editing their own genes, or indeed getting their genes edited by somebody, have been very simple examples where they've not really tried to do too much at once because obviously they were scared they would mess it up if they did. And what they have done hasn't been particularly easy to verify most of the time, except for the cases where they've tried to eradicate leukemia and succeeded with that.
Yeah, leukemia is like they use modified HIV to do it. There's also that cystic fibrosis gene editing thing that's in cystic fibrosis gene modification one that's in Europe at the moment.
That's a new one to me.
They basically looked at the cystic fibrosis gene, found it's a small point mutation that they could put into the gene to actually make it stop making the bad product, and introduce that in with the virus. You basically take an inhaler of the virus.
Yeah.
And modifies your lungs for a while until your body kicks out. Then you just take another inhaler. Actually worked really well. And
Dreamcloud, I do want to circle back around to Dreamcloud. Dreamcloud, did you have a question?
Yeah, it was sort of another off-topic type of thing, so I didn't want to drag the topic off in a different direction if it wasn't.
we're already well away from the topic.
Okay. So I guess this is sort of the gene engineering is, so when you want to change something in someone's DNA, you send something in to change it. But if you want to make that change, you'd have to change literally every single cell that uses that part, right? How do you guarantee that? Like how you, like, oops, I accidentally changed only half of your cells into so-and-so.
That comes down to like a lot of medicine. So a lot of things like you take antibiotics and things to kill bacteria. You have to kill all the bacteria, otherwise it doesn't really work. So there is that kind of level of there are like mathematical models you can test, you can see how far you've gone. There's no real way to guarantee. The general idea is you would give somebody enough of whatever does your modification to do the thing, or give it over a long enough time to do the thing. And then to actually do that, you would need to be very clever with your measuring and monitoring and things to do it.
In terms of like whole body gene edits, one of my previous labs at Boston Children's Hospital, one of the techniques we're looking into was a blood perfusion method of delivering viruses, along with a little bit of a thing to enable like actual cell invasion with that. And I wasn't on that project. I was just like kind of aware of it, and they got pretty reasonable rates of infection. It was still like very much heterogeneous and dependent on cell type. And it was still like with a baby AAV, or a baby virus being AAVs, and those would not be good enough for most purposes in terms of what we're thinking. But even so, like they were able to get a lot of infection. Sorry, Sai, what?
Yeah, the AAVs are way too small for the kind of things we would be doing.
Yeah, they're good for like one gene and a small gene at that.
And I was just going to say, this is the kind of thing that I've been working on for quite a while, and I expect to work on for a long time yet. And it's going to probably involve a need to get some really complicated simulations done as well, so as to avoid having to go through quite so many iterations of experiments on living things. But in the end, I think it should, in very sort of loose theory, be possible to be able to edit all the cells' genes in the body. It's just going to take a lot of careful thinking and really clever design.
Yeah, well said.
Sort of gene editing vectors you would need.
Yeah, exactly. Well said. And it's also worth pointing out. Sorry, it's also worth pointing out that you don't necessarily have to get all the cells in the body, just the ones that matter. And you would use math to figure out what are the ones that matter.
Well, the thing about, like we were saying about the cystic fibrosis treatment there with, was it lentivirus?
Might have been lentivirus.
Yeah, one of the sort of traditional viral vectors, so to speak, anyway. The thing about that is obviously you're only able to edit some of the cells. You're never going to get them all. And even if you got all of the cells in the lungs, it's only the ones in the lungs that you're editing and not anything else around them. And so, yes, you're able to, for a time, solve the problem of, the cystic fibrosis genes. But after a while, as those cells get renewed and replaced, the cells that produce the replacement cells don't necessarily have the edited genes in them. And so you're putting back the original problem again, just by the process of time and the cells being renewed. So then, like you said, you'd have to take another inhaler to keep on top of that. So you've not really permanently cured it.
Yeah, it's not permanently cured. There's a general approach in kind of things like medicine. When you're messing with humans, you generally want a conservative approach. You do as little as possible to do what you need to do, particularly with gene editing, because if you hit the wrong gene, you could be in big trouble.
Yeah, which is why we need to make sure that
for gene modification is just we can't hit what we want to hit all the time.
Exactly. So that's why we need to make sure that we have a really, really sort of accurate system of hitting the right genes every time. And then we need to work out how we can get it to all of the cells, so that we're not just seeing our edited cells being replaced by unedited ones.
Yeah.
One other important thing to call out is that gene editing isn't necessarily the only way to accomplish our goals either. It's a great one, but it's tough, and we know that. So
yeah, like I said, you've got all this crosstalk, you've got chemical changes, etc. Biology is very complicated. Engineering is a lot more simple because you can isolate problems. I favor engineering because I work in biology. It keeps going wrong for me, and I'm doing simple things. But that's me. Aether likes biology. We scientists differ in things.
Are there times where you could combine these two or two different methods to maybe try and get the desired effect? Like maybe combining bioprinting with genetic brain fart.
Genetic brain farts sound a little bit unappealing.
Yeah, they do.
Yeah. So combining techniques is not. Yeah, we're not excluding that. That's just we tend to talk about the techniques in isolation because that's how they're done at the moment. But like gene editing and human cells and things, you use a bit of chemistry to do it because you can encase your foreign DNA in membrane bubbles, and that needs a bit of chemistry to work, and then that helps you do your gene editing much better in human cells. So there's a lot of this interdisciplinary stuff. It's just if you've got, so we kind of don't quite know how to do it in biology. We don't know how to do it with bioprinting. If you try and combine them right now, you just end up with a lot more unknown. So yes, it's definitely something you would look into, but it's currently the speculation goes off a cliff, kind of.
Right.
But yeah, it's definitely something. Oh, yeah, good idea, Arthur.
Yeah, I just thought I'd put that in there as a kind of footnote to it.
Yeah, it's just, yeah, it's definitely durable. It's just they were speculating about stuff that hasn't been invented yet, so we'll have to see.
That's cool.
All right, are we? Do we have any other questions?
Oh, good. All right, everybody. Thanks for coming. Thanks for
Thank you
hanging
for
around,
listening.
asking questions. Well, asking us questions, actually.
Yeah, that
It's
was
been
awesome.
informative. Thank you.
Yeah, thanks, Si.
Might do it another time. We'll see. I'll keep you guys informed if we do another one.
Yeah, thanks very much for putting together this presentation, and I look forward to doing these again.
All right, catch you guys later.
See ya.
Yeah, see you.
Have a nice day.